Hello everyone and welcome to Instant Biology by Dr. Neelab. Today's lecture is about complement system. This is a new lecture in the subject of immunology. This lecture would be divided actually into two parts. In the first part I would be dealing with the concept of complement system, what is complement system and what are its constituents, how the naming has been done of complement system. We would be talking about these type of things. In the next lecture or part 2 of this video, we would be talking about what are the pathways through which this complement system is activated. So first of all, in this part, let us learn what is complement system. So this complement system was actually termed by Paul Ehrlich. Now this has a peculiar name, right? Complement system. Why? Because it is complementing your immune system, your normal defense system. It is complementing. That is why it is called as complement system. Now let us come to the components of complement system. There are multiple components of this complement system. The first thing is that there are multiple proteins, right? Some are serum proteins, whereas some are cell surface proteins. Okay, these proteins are components or parts of complement system. Now these proteins can also be of the nature of glycoprotein. What does glycoprotein mean? Glycoprotein means whenever a carbohydrate is attached onto the surface of a protein, then that protein is called as glycoprotein. So again, coming back to the second point, it says that there are approximately 30 components of the complement system, 30 proteins. They can be serum proteins in nature, they can be surface proteins, they can be glycoproteins basically. Okay, so next point that is important is that it is the component, or basically it is helping or it is aiding both the innate immunity as well as the acquired immunity, right? It is often asked in different life science examination that which type of immunity does the complement system belong to. So basically it is aiding in both the types of immunity. One is the innate immunity, you might be knowing about that. The other is the acquired immunity. Now again, acquired immunity is also, innate immunity is the natural, basically it is from birth, right? Innate immunity is from birth, but acquired immunity is acquired during the course of life of an individual. Now acquired immunity can be of two different types, we already know that. If you have not seen my previous videos, I would recommend you can see those videos and get a clear understanding about that as well. So acquired immunity is of two types. One is the cell mediated immunity and the other one is humoral immunity. This humoral immunity is basically made, or it is governed by your antibodies, right? So this complement system is basically aiding the humoral immunity. It is helping in the antibody mediated acquired immune response. This is the third point that I have mentioned, that this complement system is a participant of the innate immunity also, it is a participant of the antibody mediated acquired immune response also. Let us come to the functions. Now this complement system has a vast variety of functions, the primary being lysis of the cells. Now you might be knowing that our body gets infected or it gets attacked by different pathogenic organisms, right? Now the purpose of the immune system or the purpose of this complement system is to save our body, to provide a defense mechanism against these pathogens. So one of the methods through which this complement system provides a defense mechanism is lysing the pathogenic cell. It lyses it, breaks it apart. Next is opsonization. Opsonization means basically covering or basically creating a what do you say, it is not allowing the pathogen to leave. Okay, it is not allowing the pathogen to escape until and unless it is phagocytized, right? That is called as opsonization. Next is viral neutralization. What does viral neutralization means? That means we are rendering the virus inactive. Okay. Next is inflammatory responses. What does inflammatory responses means? You might be knowing that inflammation, suppose you have a wound or suppose you have an injury, you were playing something and you fell down. Because of that you have gained some injury. Now at the point of the injury there is redness, there is a higher temperature. Now these are all symptoms of inflammation. That means this complement system is also causing certain inflammatory responses. Next is clearance of immune complexes. What does that mean? Now, at times antigen antibody complexes form some very tight or very firm complexes. Those cannot be solubilized or those cannot be eliminated normally in normal conditions. So what does complement system do? It helps in elimination or clearance of these immune complexes. So this was the last point of this particular slide. Now let us come to the next slide, which is about the components of complement system. What are the components of complement system? As I've already said in the previous slide, that there are multiple proteins that are present as the part of complement system, that are parts of complement system. What are these complement proteins? What are these proteins? Now these proteins, basically these are groups of proteins, or they can be glycoproteins as well. As I have said, that there are around 30 proteins. They can be glycoproteins, they can be normal proteins, they can be serum proteins, they can be they can be surface proteins. What is the criteria, or how the nomenclature or how the naming has been done of these complement proteins? Now different proteins have been given different names. First, for example, the naming system of using the numbers, for example C1, C2, C3, C4, C5, C6 up till C9, some numbers have been given like this. That means C for complement, right? So suppose complement protein 1 has been given as C1 name, complement protein 2 has been given C2 name. This is sheerly in accordance with the sequence in which they were discovered, right, rather than in the sequence in which they are used in the complement system. They have been named just according to the sequence in which they have been discovered. Apart from C1, C2, C3, other names have also been given. For example, alphabetical alphabets have been used in order to name the complement proteins. For example, letter D, letter B, things like that have been also used. Next, trivial names have also been used. Trivial names means the names that are used in general language. For example, homologous restriction factor is one such example. Now coming to the point that where are these proteins synthesized? Now these proteins, these complement proteins are synthesized in liver hepatocytes. Liver hepatocytes means these are synthesized in the liver cells. Next, monocytes. Monocytes you might be knowing, again they are the defense cells or the immune cells, a type of immune cells. Tissue macrophages, again a type of immune cells. Epithelial cells of GI tract, gastrointestinal tract, right from the buccal cavity or the esophagus or the mouth till the anus. This entire region, this entire GI tract, the epithelial cells of this tract are basically synthesizing this complement system. Now the major synthesis takes place basically in liver only. Now coming to the concentration, what is the concentration of this complement proteins in your body? Around 3 milligram per ml of blood is the concentration of the entire complement system. That means you can sum up all the complement proteins and then only you would get the concentration of 3 mg per ml. But there are certain complement proteins that are exceedingly higher in concentration as compared to others. For example, C3 is majorly present. That means it has a very high concentration as compared to the other complement proteins. It has a concentration of 1 mg per ml if you compare it to the other complement proteins. Now this was all about the second slide or this slide. Let us move on to the third slide. Now what does this third slide say? What are complement activation pathways? Now you know that complement system is one of the systems that is switched to at the last, because when you do not have any option left, then only the complement system is switched on. Okay, otherwise it is generally in the switch off condition. That means all the complement proteins are inactive at the beginning. So they are activated only when there is certain need of activation. When they are present in the inactive form, they are called as proenzymes or zymogens. So this is a general term you might be knowing. Whenever something is inactive, it is called as proenzyme or zymogen, right? Next, when it becomes activated, basically what happens is one component activates another component, another one activates another component, another one activates another component. So basically it is a cyclic or sequential event that goes on happening, one component activating other one, other activating another one, another one activating another one. So this goes on happening. So this is a sequential event. Next is activation causes splitting of components. That means suppose a complement named C3 gets activated. Okay, suppose a component called as C3 gets activated. Now when C3 will get activated, it will get broken down. It will get broken down into two parts. One will be the smaller part, other one would be the larger part. Let us name them as C3A, C3B. Okay, C3A, C3B. Now which one will be called as C3A and which one will be called as C3B? This is important to understand. The smaller fragment is often called as A. That means C3A would be the smaller fragment, C3B would be the larger fragment. Okay, but this rule has an exception. That rule that the smaller fragment would be A and larger fragment would be B, this rule has an exception in case of C2. That means in case of C2, when the C2 will be broken, C2A would be the larger fragment, C2B would be the smaller fragment, just reverse of what we just learned in the I believe second point. Okay, now one more important thing is that out of these two components, one component will stay at the site which got it activated, right? One component amongst the two, A and B, would stay at that place and the other component will move at a different position or move outside or move away. In general, always remember that A component will move away. Now this I'll give you a trick over here. That means A for away. Okay, whenever you say A for away, that means the A component, for example C3A, will move away. Why it is moving away? What function does it have? What will it do when it moves away? What is the reason? Because in the first slide only I said that it is mediating the inflammatory responses. Okay, so the one that moves away, it actually is going for recruitment, or it is going for conducting the inflammatory responses only. Okay, and what about B? B will stay at the target site. It will get bound to the target site because of which it got activated. So in this slide also I've highlighted A means away, it is responsible for the inflammatory responses. B means stay, it will bind to the target site. Okay, this is what I was saying. Now let me just move myself away so that you can read properly. So for inflammatory responses, A and B binds to the target near the site of activation. This was the concept. Okay, now let us get back to the slide. This slide is completed. Let us come back to the next slide. Now to this rule also there is an exception. What is the exception? C2 again creates an exception, because in C2 we have already studied an exception, that C2A and C2B. In general we said the A part is smaller, B part is larger, but in case of C2 we have already said that this rule will be reversed. C2B would be the smaller protein, C2A will be the larger protein. Similarly, there is an exception with the away concept and staying concept also. That means C2B over here will move away, whereas C2A will stay. Okay, and what will C2B do? It will go for the inflammatory responses. Now coming back to the complement fragments. Okay, what does complement fragment means? Actually the complement systems would not be able to perform functions individually. So the criteria or the way in which the complement system basically functions is that they join together to form complexes. Okay, they would not be able to do individually, they would not be able to function individually. Suppose C3b is combining with factor B, it is combining with factor D or something like that, then only it can function. There is a cumulative function. They can do function only when they are combined together. Okay, so they interact to form functional complexes. This is the second point. And next is, whenever they are combining like this, they gain functionality. They start doing functions. Okay, so for example, C3b, capital B, small b, things like that. When one more important thing, whenever a component or whenever a complex gains function, how do you represent it? You represent it by a straight line just above the name of the complex. For example, in over here you can see that C3 small b, capital B, small b, it is having a bar just above it. That is being represented because it is activated now. It has gained function now. That is why we are representing it in this way. Now, there are three ways in which a complement system can get activated. What does that mean? Remember that this complement system is our last resort, right? So we have to use it very judiciously. That means you cannot use classical system, you cannot use complement system in an everyday process. It will be used whenever there is a very there is a sheer need of existence only. It is essential to your survival. Then only complement system gets activated. Now there are three possible pathways through which you can activate your complement system. These are classical pathways, alternative pathways, and lectin pathway. I would be talking about these three topics in the next or subsequent lecture. Okay, so this was a brief overview about what is a complement system, how it functions. Now, before ending this particular video, I would like to just inform you that for those students who find bioprocess engineering tough, I have prepared a book that is just rupees 215 inclusive of your dispatch charges. It will be very helpful for your GATE biotechnology examination. Okay, and it comprises of the complete unit number 4, unit number 5. Okay, so you can get in touch with us if you want to purchase the book. Plus the live classes for the bioprocess engineering batch are starting from 5th November. The total cost is rupees 1400 only. That means the bioprocess engineering class would cost rupees 1400 only. It is for 1 month. I have already posted the video link for that, and the charges for the book is rupees 200 in this case. Otherwise the book is rupees 215. If you are in Lucknow, you can pick up the book at a much lesser price, that is rupees 165 if you are in Lucknow and you want to pick it up from our office. Otherwise, if you want to take the live classes as well as the book, the price would be rupees 1600. With this, I would like to take your leave, and plus I would request you to subscribe to my channel, to like the video, and do not forget to press the bell icon and share with your friends. Thank you so much and have a great day.